There's a lot of interest about the heritability of autism, and specifically its genetics. But most folks may be unaware of the wide variety of different types of genetic variations that can be playing roles in autism susceptibility. Before I started studying genetics, I myself had a very limited grasp of mutations, usually picturing single point mutations in the DNA as the major form of genetic variability across humans. And truthfully, this is usually what we learn in most basic undergraduate biology classes, whereas in fact, single point mutations are just the tip of the iceberg. Now, this video is going to be at least a two-parter. Today, I'm going to talk about the basics of human genetics, but in the next video, I'm going to explore the types of genetic variations we see in the autism spectrum. So stay tuned for part 2. Okay, so Genetics 101. As an aside, even though they find it a bit strange to get a genetics crash course in my introductory neuroscience class, I do love to torture my neuroscience students a little bit with some basic genetics because it allows them to understand the different conditions we explore in my class better, like autism, Parkinson's, and migraines. Without this basic understanding, it's much harder to appreciate the genetic complexity of many of these conditions. So with that in mind, let's dive in. First off, the human genome is of course made up of DNA, and it's over 3 billion nucleotides long. These nucleotide bases are adenine, cytosine, guanine, and thymine, but we usually just refer to them by their abbreviations, A, C, G, and T. Now, that DNA, when it's not in use, is wrapped around these large protein bundles called histones, a little bit like a string when it's wrapped around a yo-yo. This is a space-saving measure, as well as a means to keep genes locked down when they're not supposed to be expressed. When we talk about genes, most of the time we're referring to protein coding genes. These are segments of DNA that, when expressed, are transcribed into an intermediate RNA, which is eventually translated into a protein. Proteins do a lot of things in your cells, like helping to support the structure of the cell, promoting chemical reactions, or sending signals to other cells. Now, only about 1% of your entire genome is made up of these protein coding genes. The rest of the genome is referred to as non-coding DNA. And contrary to its name, a lot of it is actually transcribed into RNA, which performs some of its own unique functions in the cell. Some of the non-coding DNA that isn't transcribed into RNA also helps to regulate genes, determining whether they get expressed, when, and how. All of this extra regulatory material allows your genome to behave in very complex ways. For instance, it allows different combinations of genes to be expressed across different cell types at different times in cells' lives. In addition, it can also regulate which version of a gene gets produced. FYI, although there's just a single blueprint per gene per chromosome in a cell, very different versions of that gene can be produced based on how its RNA gets spliced. These different versions can then be used to do different things in the cell. For instance, if version number 1 of an imaginary gene A may be relegated to function within the nucleus of the cell where the DNA is kept, but version number 2 of that same gene may be able to shuttle past the nuclear envelope, which is the membrane that surrounds the nucleus, and work its magic out in the cytosol of the cell. This can change what that protein is doing and how it's doing it. Now, a lot of these protein coding genes are pretty well conserved across different animal species. They don't like to change a whole lot over time. So evolution has instead focused a lot of its efforts on evolving the non-coding regions of the genome, allowing more and more nuanced control over how and when genes are expressed. A lot of the differences we see between ourselves and our closest living relatives, the chimpanzees and bonobos, are largely thanks to differences in our non-coding DNA. Otherwise, our coding DNA is roughly 98% identical. And as an aside, if you've ever heard that humans and chimps are 98% genetically identical, it's referring to these protein coding genes, not the rest of the genome. So that's definitely a stretch of the biological truth. Okay, so what about mutations? Mutations happen all the time. We're constantly collecting mutations in our cells over our lifetimes, which, when they turn cancerous, they either selflessly commit cell suicide, or if not, hopefully our immune systems manage to snuff out the ones that go rogue. But we also frequently have mutations in our sex cells, i.e., sperm and eggs. Now, about 1/3 of all human conceptions are miscarried as a result of major gene mutations that aren't compatible with life. So even though we tend to think of major and really nasty mutations as relatively rare, that's only the case in people who have made it to birth and beyond. But what types of mutations are we talking about? What's the range of mutations that can occur during development? Now, mutations can actually vary tremendously in size. They can range from whole genome duplications, which are also known as polyploidy. Now, whole genome duplications are exactly as they sound. They're extra copies of the entire genome. So rather than having 2 copies of the genome, one from mom, one from dad, an individual may have 3 or more copies. Now, for humans, having an extra copy of the human genome in all the cells of their body is not conducive to life, and it unfortunately leads to miscarriage or is lethal shortly after birth. So this is not something that we see in adult humans. Now, we do see it in certain normal tissues within our bodies that have extremely high metabolic demands, like certain cells of the liver and in the placenta. This is completely normal, and we all have this, and this is mainly because extra copies of the genome can actually be really useful for a cell that has to make a lot of RNA and proteins continuously. Unfortunately, for this same reason, we sometimes see whole genome duplications in cancerous cells too, because it helps support their really high metabolic needs, even though that process is considered pathological. Now, another type of large-scale mutation is having too few or too many of a particular chromosome, which is also known as aneuploidy. We see this, for instance, in most people with Down syndrome who have 3 copies of chromosome 21, otherwise known as trisomy 21. I'll talk more about Down syndrome in the next video as well, since we see about 10 to 15% of people with Downs also have autism. The next size down are large chromosomal abnormalities that don't affect the entire chromosome, but instead target a large portion of one. For example, a whole arm of a particular chromosome may be missing, it may be duplicated, or it may even be inverted or translocated. Now, an inversion means that you've taken a portion of a chromosome, snapped it off, turned it around, and stuck it back on the chromosome in the reverse orientation. Meanwhile, a translocation means you've taken part of a chromosome, snapped it off, and stuck it on a different chromosome, or sometimes on the other end of the same chromosome. Often a translocation involves the trading of genetic material between 2 chromosomes, so you'll usually see 2 chromosomes that are affected. Inversions and translocations, even though you're not technically missing genetic material, in a different orientation or chromosomal location, they can still cause problems for the regulatory machinery that controls how and when genes on that section of the chromosome are expressed. Often, as a result, many of those genes don't get expressed properly. The next mutation size down are smaller insertions and deletions, often referred to as indels. Indels range anywhere from 1 nucleotide in length all the way up to roughly 5 million base pairs. The larger indels are often referred to as copy number variants, or CNVs, which you may have heard of CNVs in relation to autism, since these are types of mutations that are sometimes found in autistic people who have a new or de novo mutation that isn't shared by mom or dad. Most typically, CNVs are deletions or duplications of material ranging from 1,000 to 5 million nucleotides long. Smaller indels, which are sometimes called microindels, can also happen, and these typically range from 1 to 50 nucleotides in length, and again, genetic material is usually either deleted or duplicated. Finally, the last type of indel that we may see are repeat expansions and retractions. These are typically sequences of repetitive DNA that have a tendency to expand or contract in numbers of repeats because these repeats are prone to instability. A classic example is the long string of CGG trinucleotide repeats in the promoter region of the FMR1 gene. Now, a promoter is basically just the on-off switch for a gene, and in the promoter of the FMR1 gene, there's a long trinucleotide repeat track that's made up of the nucleotides CGG repeated over and over and over again. Normally, most of us have anywhere between 26 and 32 CGG repeats in this FMR1 promoter. But sometimes, thanks to the repetitive nature of this DNA track, this unstable region can increase in length, adding more and more CGG repeats to the promoter. And the longer the repeat track, the more unstable it is. When this happens, if the repeats occur anywhere between 55 and 199 trinucleotides long, the person will have what's known as Fragile X premutation. And if a person inherits 200 or more repeats, they will most likely develop Fragile X syndrome. Fragile X syndrome has very high rates of autism. About 1 in 2 males and about 1/5 of females with the condition will also be on the autism spectrum. Since the FMR1 gene is on the X chromosome, that means that males tend to be more severely affected than females because females have a spare X chromosome. So not all the cells in women's bodies are going to be affected by the mutation. This is thanks to a normal process called X inactivation. Since only a single X can be safely expressed in a cell at a given time, in females and in the rare male who has an XXY genotype, otherwise known as Klinefelter syndrome, a single X chromosome is randomly turned off. Therefore, on average, FMR1 mutations are only expressed in about half of the cells in a female's body, whereas in males, because they only inherit a single X chromosome, all their cells are affected. This can mean sometimes that women with Fragile X syndrome can go undiagnosed, or they may not get diagnosed until they themselves have a son with Fragile X syndrome. Okay, so the last type of genetic variation I'm going to talk about are single nucleotide variants, or SNVs. Although they're just one nucleotide long, they're not the same thing as a microindel because you're not adding or deleting material. Instead, an SNV just converts one nucleotide to another. So for instance, there may be a point in the DNA that used to be an adenine or an A, and it mutates into a guanine or a G. Sometimes this happens in a segment of the genome that codes for a protein, otherwise known as the exome. When a nucleotide in the exome is mutated, one of 3 things are going to happen. 1, the mutation doesn't result in a change in the protein sequence. This is known as a synonymous mutation. 2, the change does alter a single amino acid in the protein, and this is called a non-synonymous mutation. And by the way, amino acids are just the building blocks for proteins. And finally, 3, the change results in a premature stop, and the cell fails to produce the elongated protein, which means the protein may not be able to do its job. Now, you may have also heard the terms single nucleotide polymorphism, or SNP, SNP. And for the most part, these are just single nucleotides in the DNA where humans have a tendency to vary somewhat from each other. Since the vast majority of our genomes are identical, that means only a comparative handful of sites are what we would call polymorphic. For comparison, of the 3 billion nucleotides in the human genome, only about 90 million of them show variation across the human species, and about 10 million of those occur in at least 1% or more of the human population. So, in other words, about 1 in 10 polymorphisms are common. The other 80 million-ish are rare. Okay, I think that's enough for our introductory lesson on human genetics. Next week, I'm going to be doing part 2, where I'll talk about the different kinds of genetic variation we tend to see in autism, ranging from extra chromosomes to copy number variants to common polymorphisms. So stay tuned for our next deep dive down the autism rabbit hole. Thanks for watching, and ciao.